Biology of hair follicles. Evolution and function of hair. Primary purpose of hair in mammals are insulation and protection from the elements. Primary purpose in humans is influences social interactions. Hair-related conditions such as alopecia, hair loss, and hirsutism, hypertrichosis, excessive hair growth, cause significant psychological distress and support a multi-billion-dollar industry. Hair follicle as a model for studying a range of biological processes including cyclical regeneration, developmental biology, stem cell biology, keratinocyte and melanocyte stem cells, and epithelial-mesenchymal interactions. Hair follicle embryology. Hair follicle development occurs in 8 defined stages, progressing from placoid formation to a fully mature follicle. Much of what we know about follicle development comes from studies in fruit flies, where conserved genetic pathways are shared with mammals. These include BMP signaling, hedgehog, Notch, WNT, and FGF pathways, all playing critical roles in follicle induction, morphogenesis, and cycling. Hair follicle formation begins cranially and then spreads caudally. The earliest hairs formed are lanugo hairs, which are thin, non-pigmented, and provide a temporary protective covering for the fetus. These are usually shed before or shortly after birth. Homeobox genes direct the spatial arrangement of follicles across the body surface, ensuring even distribution and pattern formation. Any disruption can lead to patchy or abnormal hair growth patterns. Specific genes define hair type and quality. For example, FGF5 mutations can cause excessive hair growth by prolonging the growth phase, while EDAR variants in humans are linked to hair thickness differences across populations. Molecular regulation of hair follicle morphogenesis. This schematic summarizes the molecular regulation of hair follicle morphogenesis. Growth factors like BMP, EDA, HGF, KGF, and SHA provide inductive signals. Their receptors, such as BMPRIA, EDAR, EGFR, and PTC1, mediate epithelial-mesenchymal interactions essential for follicle initiation. Adhesion molecules, including ecadherin, pcadherin, and NCAM, regulate cell polarity and tissue organization. Transcriptional regulators such as FOXN1, GATA3, LEF1, HOXC13, and LHX2 orchestrate lineage commitment and differentiation. Together, these pathways tightly control each stage of follicle morphogenesis, ensuring proper hair patterning and growth. Hair follicle development stages. Hair follicles begin with epithelial placodes, interacting closely with dermal papilla cells. This epithelial-mesenchymal communication is key to initiating follicle formation. As development progresses, the epithelial germ elongates into a bulbous peg. The outer root sheath differentiates into 2 bulges. One becomes the sebaceous gland, while the deeper bulge provides stem cells critical for ongoing hair cycling. Molecular regulation of development. Hair follicle development relies on a fine balance of activating and inhibitory signals. WNT beta-catenin primes epithelial cells for follicle formation. EDA interactions further drive early development, while BMPs inhibit follicle induction unless blocked by noggin. SHH signaling is indispensable for follicle morphogenesis. Knockout models show poorly formed follicles. Mature hair follicle and hair types. A mature follicle is highly organized, with root sheaths encasing the hair shaft. Hair types vary by size and structure. Lanugo in fetuses, vellus over much of the body, and terminal hairs in scalp, beard, and other androgen-dependent sites. In androgenetic alopecia, terminal hairs progressively shrink into vellus-like follicles, leading to thinning of hair. Follicle anatomy and structure. The hair shaft consists of 3 layers, while the follicle contains specialized compartments. The dermal papilla at the base orchestrates growth, while the hair matrix produces the shaft and root sheath. The bulge region is particularly important, housing stem cells that enable regeneration. The erector pili muscle provides piloerection, the goosebump response. The accompanying figure illustrates follicle anatomy and cycling phases. This picture shows hair cycle and anatomy. Hair follicle cycle. The hair follicle cycle is continuous and asynchronous. Anagen drives growth through matrix proliferation and shaft emergence. Catagen halts mitosis, initiating apoptosis and upward migration of the papilla. Telogen is a resting phase culminating in a club hair, and exogen marks active shedding. This cycle is tightly controlled by multiple growth factors and signaling pathways, such as KGF, FGF5, TGF-beta, and EGFR. Hair pigmentation reflects melanin production by melanocytes, tightly coupled to the hair growth cycle. These melanocytes originate from neural crest-derived melanoblasts, guided by PAX3-MITF. Ednerb and c-KIT. In adults, melanocyte stem cells in the bulge replenish pigment-producing cells during anagen. Their regulation relies on TGF-beta, Notch, and BCL2, with depletion leading to progressive graying. Two pigment types are synthesized, eumelanin dark and pheomelanin light, controlled by MC1R signaling. Alpha-MSH favors eumelanin, while ASP in mice skews towards pheomelanin. With age, follicular melanocyte numbers decline due to stem cell exhaustion, explaining the transition to gray or white hair. This schematic illustrating localization of distinct subpopulations of melanocytes and dynamic of the follicular melanocytes during anagen. Conclusion. The hair follicle is a highly dynamic mini-organ formed through intricate epithelial-mesenchymal interactions. Many developmental signaling pathways, including WNT, SHH, BMP, and Notch, are reused in this process. The follicle's unique architecture allows continuous cycling. Crucially, epithelial stem cells and melanocyte stem cells ensure follicle renewal and pigmentation, highlighting the follicle as a model system for studying stem cell biology and regenerative medicine.